Welcome to cell transport. Cell transport is the movement of material across the cell membrane. The cell membrane is a phospholipid bilayer, which is the organization of phospholipids in a membrane with the hydrophilic phosphate group heads facing outward and the hydrophobic lipid tails facing inward. In this diagram, the circle represents the hydrophilic head and it's on the outside because they're water-loving. Hydrophilic means water-loving. On the inside is the hydrophobic tail, water-fearing. All of these phospholipids end up forming the cell membrane. Now let's look at the types of transport: active and passive. Active transport requires energy. Passive does not. Before we go in depth with the types of transport, we have to know what a concentration gradient is. It's a difference in concentration on both sides of a membrane. One side is always higher than the other. So now active transport. This is the gradient, and these are the particles moving upward. So you can say that they move against the concentration gradient since they move from low concentration to high concentration. All active transports require a protein because it requires the cell to perform work. Protein pumps are channels that move ions, charged particles, or small polar molecules, which are no larger than a monomer, against the concentration gradient. Requires energy. An example of a protein pump would be a proton pump, which creates a high concentration of protons. Endocytosis is how cells bring in large molecules. In the following diagram, the molecules are trying to enter the cell, and then the cell is engulfing it. Once it engulfs it, the phospholipids branch off and form a vesicle around it to bring it into the cell. There are two main types of endocytosis: phagocytosis, which is engulfing large material or cells and then digesting them with the lysosome. So you can think of it as food. Then there's penocytosis, which is engulfing a large amount of fluid with many things dissolved in it, like water. Exocytosis is how cells release large molecules. In the following diagram, the vesicle with the molecules is exiting the cell, and then it just connects with the membrane. In the third image, you can see that the vesicle isn't being released, it's just the molecules. Since the vesicle merges with the phospholipid bilayer, exocytosis makes the cell grow larger. Passive transport is the opposite of active. Particles move down the concentration gradient. Simple diffusion is a type of passive transport. Simple diffusion is a movement of particles from a greater concentration to a lesser concentration until they're equal, or you could say until they reach dynamic equilibrium, which is zero net movement. Small and non-polar molecules can diffuse directly across the cell membrane when they're moving down the gradient. Facilitated diffusion. Diffusion of molecules through protein channels and carriers. Small and polar molecules like glucose or H+ ions need protein channels to travel across the cell membrane when they're moving down the gradient. Because the cell membrane is non-polar and does not like to interact with polar molecules. Facilitated just means to make easy of, so it helps the molecules move faster. What increases the rate of diffusion? Greater difference in concentration, higher temperature, and shorter diffusion. Osmosis is the diffusion of water, and there's two types in this too. Facilitated. Since water is polar, it moves faster through a protein channel called an aquaporin. The aquaporin makes moving water easier, which is why it's under the facilitated. Simple. Water can move slowly through the phospholipid bilayer because there's just so much of it. As you can see, facilitated is much faster and simple is slower. These are key terms you need to know. Hyper is just higher, hypo, lower. Iso, same. Those are pretty straightforward. Then there's tonic, which is the concentration of solute. Cell refers to the inside, solution refers to the outside. That's an easy way to decode any problem that you get. So for example, this a hypotonic cell is placed in a hypertonic solution. This white box represents the solution, and the cell on the inside is that circle. Since the solution is hypertonic, it means there's a higher concentration of solute, and the solution is outside, meaning in the white box. That's why there's so much of the solute there. On the inside, there's a lower concentration of solute, since it's a hypotonic cell. That's all you need to know for cell transport. Thanks for tuning in, and I hope you understood the lesson.